What actually is cholesterol? The text calls it an amphipathic lipid, which sounds complicated.
It sounds like it has a split personality, and chemically, it kind of does. Amphi means both.
Mm.
So you've got this big, bulky, rigid four-ring structure, the steroid nucleus, and a floppy hydrocarbon tail.
Okay.
That entire part of the molecule is hydrophobic. It despises water. It wants to get as far away from the watery environment of your blood and your cells as possible.
But there's another side to it.
There is. There's this tiny little hydroxyl group, just an oxygen and a hydrogen tacked onto the other end. And that little group is hydrophilic. It loves water.
So why does that matter? Why is the split personality so important?
Oh, it's everything. It's the entire basis for its main job. Because of that dual nature, it can slide perfectly into a cell membrane.
How does that work?
It orients itself so that the tiny water-living head points outwards, towards the fluid inside and outside the cell. And it buries that big water-hating tail deep inside the fatty, oily part of the membrane.
So it's like the mortar between the bricks of a cell wall.
That is a perfect analogy. The text mentions it regulates permeability and fluidity, and that's exactly what it's doing. It's a stability buffer.
A buffer?
Yeah, think about it. Without cholesterol, your cell membranes would be incredibly unstable. If things got too cold, the fats in the membrane would pack together, crystallize, and the cell would shatter.
And if it got too hot?
The membrane would basically melt. It would become too fluid, too leaky, and the cell would just fall apart. Cholesterol wedges itself in there and stops the fats from getting too close or too far apart. It keeps the membrane strong but flexible.
So without it, we'd literally fall apart.
Complex life as we know it would be impossible. So job number one is structural. It's holding us together.
But looking at the list of its other jobs, it's not just a structural component. It seems like it's the ancestor of a whole dynasty of important molecules.
It is the patriarch of the entire steroid family. And this is the part that gets completely lost in the public health messaging. If you could magically snap your fingers and make all cholesterol disappear,
what happens?
well, you'd immediately lose all your corticosteroids.
Which means no stress response.
No cortisol to manage your metabolism and blood sugar. No aldosterone to manage your blood pressure. Your ability to control inflammation, gone.
Wow. And the other hormones?
All the sex hormones are gone. Testosterone, estrogen, progesterone, they all start as cholesterol. The entire reproductive system would just shut down.
And what about vitamin D?
Vitamin D, too. The process starts with a derivative of cholesterol, 7-dehydrocholesterol, that sits in your skin. When UV light from the sun hits it, that's what kicks off the synthesis. No cholesterol, no vitamin D.
And then your bones start to demineralize.
Exactly. And maybe most immediately, you lose all your bile acids.
Which, as we'll get into, are critical for digestion.
Critical is an understatement.
Yeah.
Without bile, you cannot digest fat. You'd suffer from severe malnutrition no matter how much you ate.
So when we call it a villain, we're really ignoring that it's keeping us alive on like 5 different fronts simultaneously.
It's absolutely essential for life, period.
Okay, that point is well and truly made. So where does it all come from? I think most people, myself included, would assume it all comes from the food we eat. The eggs, the cheese, the steak.
And that's probably the single biggest misconception out there. The text makes it very clear. A little over half of all the cholesterol in your body is made by your body. De novo synthesis.
We're making our own supply.
Constantly, to the tune of about 700 milligrams every single day. Even if you ate a diet with zero cholesterol, your liver would just ramp up its own production to compensate.
Which brings us right to section 1, the factory, the biosynthesis. Looking at the diagrams in the chapter, this is not a simple process. This looks costly.
Incredibly energy expensive. The body would not do this unless it was absolutely necessary. It happens in the cytosol and on the membranes of the endoplasmic reticulum.
The ER.
Right. And while most cells can make some, the real industrial hubs are the liver, the intestine, the adrenal cortex, and reproductive tissues, places that either export it or need a ton of it for hormone production.
And the raw material, what's the starting block for this whole thing?
Acetyl-CoA.
Of course it is. Acetyl-CoA, it feels like every metabolic pathway we ever discuss comes back to this one molecule.
It's the universal currency. It's a simple 2-carbon unit that you get from breaking down carbs, fats, proteins. It's the ultimate Lego brick. And here's the mind-blowing fact from the source.
Go on.
Every single one of the 27 carbon atoms in that huge, complex cholesterol molecule comes from acetyl-CoA.
Wait, all of them?
All of them. The body is building this incredibly intricate molecular machine using only one type of tiny 2-carbon brick.
That's amazing. Okay, so let's walk through the assembly line. The text breaks it into 5 main stages. Stage 1 is the biosynthesis of mevalonate.
And if you're going to remember one single step from this entire deep dive, this is the one. It is the most important one.
The commitment step.
The absolute point of no return. So we start with 2 molecules of acetyl-CoA. An enzyme called thiolase sticks them together to make a 4-carbon molecule, acetoacetyl-CoA.
Okay, 2 plus 2 is 4. Simple enough.
Then another enzyme, HMG-CoA synthase, adds a 3rd acetyl-CoA. Now we have a 6-carbon molecule called HMG-CoA. So far, so good.
Still just stacking bricks.
But now comes the critical moment. A very special enzyme grabs that HMG-CoA and converts it into a different 6-carbon molecule called mevalonate, and that enzyme is HMG-CoA reductase.
The text highlights this one in bold. It calls it the principal regulatory step. Why is this enzyme the superstar of the pathway?
Because this is the master switch. Up to this point, HMG-CoA could have been used for other things, like making ketone bodies. But once HMG-CoA reductase hacks, that mevalonate is locked in. It is committed to this pathway.
It's on a one-way street to becoming cholesterol.
Exactly. And the cell has to spend 2 molecules of NADPH, pure reducing power, pure energy, just to make this one reaction happen. It's a huge investment.
And because it's the bottleneck, it's also the target for intervention.
It's where pharmacology comes to the party. We're talking about statins.
Lipitor, Crestor, all of them.
All of them. They are all competitive inhibitors of this one enzyme, HMG-CoA reductase.
Unpack competitive inhibitor for us. What's it actually doing?
It means the drug molecule is shaped a bit like the normal substrate, HMG-CoA. So it fits into the enzyme's active site, but it doesn't react. It just gets stuck.
It gums up the works.
It handcuffs the factor worker. The enzyme is occupied. It can't bind to the real HMG-CoA, and the entire assembly line grinds to a halt right at the very beginning.
Which is very effective at lowering cholesterol, but it leads to a potential problem, right? As we move to step 2.
A very significant potential problem, yes.
Step 2 is formation of isoprenoid units. We take that mevalonate, we pump it full of energy from ATP 3 times, and then we chop off a carbon to make these little 5-carbon building blocks.
Isopentenyl diphosphate. These are the active isoprenoid units, or like energized Lego bricks.
But the chapter points out these bricks aren't just for building cholesterol.
No, and this is the crux of some of the side effects of statins. These same isoprenoid units are precursors for other vital molecules. Dolicol, for example, which is needed for making certain types of proteins. But far more importantly, they are the building blocks for ubiquinone.
Ubiquinone. That's coenzyme Q10, isn't it?
It is, OQ10.
The thing you see in supplements for energy. The spark plug for the mitochondria.
That's a great way to put it. It's an essential component of the electron transport chain, the process that generates almost all of the ATP in your body. Your mitochondria cannot function properly without a steady supply of CoQ10.
So if you're taking a statin, which blocks the factory way back at step 1, you're not just stopping cholesterol production, you are also choking off the supply line for the molecule that your muscles need to make energy.
That is the leading biochemical theory for why a subset of patients on statins experience myopathy, muscle pain, weakness, and fatigue. You're inadvertently creating a CoQ10 deficiency in the tissues.
That is a really important connection to make. It shows that you can't just tinker with one part of this web without causing ripples somewhere else.
Nothing in biology happens in a vacuum.
Okay, so let's assume the factory is running normally. We've made our 5-carbon isoprenoid bricks. Step 3 is forming squalene.
Right now, it's just basic construction. We start snapping the bricks together. We take one 5-carbon unit and isomerize it slightly, then we connect it to another one. Now we have a 10-carbon chain called geranyl diphosphate.
Okay, 10 carbon.
We add another 5-carbon brick to that. Now we have a 15-carbon intermediate called farnesyl diphosphate.
So we're at 15.
Now for the big move. We take 2 of those 15-carbon chains and we fuse them together head to head. 2 times 15 gives you a massive 30-carbon chain called squalene.
Squalene, it sounds like a fish oil.
Well, it was first discovered in shark liver oil, so the name stuck.
Yeah.
But your body makes it constantly. But look at it. It's a long, floppy, linear molecule.
Right. And cholesterol is that rigid ring structure. It looks nothing like this. How do you get from a piece of string to a chicken wire fence?
Step 4, cyclization. And this is pure biochemical magic. It's origami.
Origami?
An enzyme called oxidosqualene. Lanosterol cyclase grabs that long squalene chain, and in a single concerted reaction, it folds it and zips it up into the iconic 4-ring steroid nucleus. The product is a molecule called lanosterol.
That happens all at once, the folding and zipping.
It's an absolutely breathtaking piece of enzymatic engineering, but lanosterol isn't quite the finished product. It's a rough draft. It still has 30 carbons.
And cholesterol only has 27.
Exactly. Linosterol is a bit bulky. It has a few extra methyl groups sticking off it that need to be trimmed.
So it needs a final polish.
That's step 5.
Yeah.
This happens in the ER membrane. It's a whole series of reactions that snip off 3 methyl groups, shift a double bond from one spot to another. We pass through a late-stage precursor called dismosterol, and then finally, we have cholesterol.
Wow. Okay, just to recap that incredible journey, we start with 2 carbon acetyl-CoA bricks. We commit them to the pathway by making mevalonate with that master switch enzyme. We energize them into 5 carbon isoprenoid units. We snap those together to make a 30-carbon string called squalene. We fold the string into a ring called lanosterol, and then we give it a final trim and polish to get to cholesterol.
And in the process, we burn through a huge amount of ATP and NADPH.
It's so expensive, the body clearly doesn't want to be doing this unless it absolutely has to.
Which is the perfect segue to the control room. Section 2, regulation of cholesterol synthesis.
Right. If the process is so expensive, you need a very, very good set of brakes. And it seems like the body has an obsession with controlling that one enzyme we talked about.
HMG-CoA reductase. It's a choke point, so that's where all the regulation is focused. The text lays out 5 separate mechanisms the cell uses to control this one enzyme.
5. That's some serious redundant security.
It shows you how important it is to not let this pathway run wild.
Let's go through them. Mechanism 1 is feedback inhibition.
The simplest one. The product of the reaction, mevalonate, and the final product, cholesterol, can both directly inhibit the enzyme. It's like a sensor on a conveyor belt. If the bin at the end of the line gets full, the belt stops running.
Makes sense. Don't make more of what you already have too much of.
Precisely.
Okay, but mechanism 2 sounds way more complex, the SREBP pathway.
Oh, this is beautiful. SREDP stands for sterile regulatory element binding protein. Think of SREBP as a manager whose job is to go to the company headquarters, the nucleus, and request more factory workers. It's a transcription factor.
So it tells the DNA to make more HMG-CoA reductase enzyme.
Exactly. But it's kept on a very short leash. It's held prisoner in the membrane of the ER by another protein called INSIG.
INSIG.
Insulin-induced gene protein. Think of INSIG as the prison guard. As long as there's plenty of cholesterol in the ER membrane, INSIG has a tight grip on SREBP. It's locked down.
So the manager is stuck in his office as long as inventory is high.
Yes, but when the cell's cholesterol level starts to drop.
Say if you haven't eaten much or you're on a drug that blocks absorption.
Right, the cholesterol leaves the membrane. This causes a subtle change in shape and INSIG loses its grip. SREBP is set free.
And it makes a run for it.
It gets escorted to another organelle called the Golgi, where it gets processed.
Yeah.
And then it marches right into the nucleus, finds the gene for HMG-CoA reductase, and switches it on. Production ramps up.
So the very moment you start a low cholesterol diet, your liver cells sense it and immediately start building more machinery to make up for the deficit.
Your body actively fights your diet. It has a set point it wants to maintain.
That's incredible. It's a constant battle. What's mechanism 3?
This one is even more direct. If cholesterol levels get very high, the cell doesn't just stop making new enzyme molecules. It actively starts destroying the ones it already has.
It sends the existing workers home.
It sends them to the shredder. That same INSIG protein helps to tag the HMG-CoA reductase protein for degradation. It's a very rapid and ruthless way to shut down production.
Wow. Okay. Now mechanism 4, the energy check. This involves AMPK.
Right. AMPK is the master fuel gauge of the cell. When your cell is running low on energy, its ATP levels drop and its AMP levels rise. High AMP is a screaming alarm signal that activates AMPK.
And what does AMPK do?
It's a kinase, so it phosphorylates things. It sticks a phosphate group onto HMG-CoA reductase.
And that turns it off.
It slams the brakes on. The cell's basically saying, look, we are in an energy crisis. We do not have the fuel to run this extensive luxury item factory. Shut it down now. We need to conserve energy for survival.
And finally, mechanism 5, hormones. This brings in the insulin connection.
Right. Insulin is the hormone of the fed state. When you eat, insulin goes up. It signals abundance. We have fuel. We have resources. So insulin activates the reductase. It encourages storage and building.
Go ahead and build some cholesterol. We can afford it.
Exactly. Whereas hormones like glucagon or stress hormones like glucocorticoids, they signal the opposite.
We need energy now.
Right. They signal a state of fasting or crisis. So they inhibit the enzyme. They shut down long-term building projects to free up resources for immediate needs.
So we've made the cholesterol. We've got 5 different ways to control its production. Now we've got a cell, maybe in the liver, that's full of it, but it needs to get to the adrenal gland or the ovaries or a muscle cell that's repairing its membrane. It has to be shipped.
And this brings us to Section 3, the logistics network.
And the fundamental problem here is that cholesterol is an oil and blood is water. They don't mix.
You can't just dump it into the bloodstream. It would clump together and form a fatal embolism. So you need a transport vehicle. You need a molecular submarine.
Lipoprotein.
Exactly. For delivering cholesterol from the liver out to the tissues, the main vehicle is LDL, low-density lipoprotein.
The Amazon delivery truck.
The Amazon truck, yes. Its whole job is to carry a cargo of cholesterol through the bloodstream and drop it off where it's needed.
And HDL is the opposite.
HDL, high-density lipoprotein, is the waste management fleet. Its job is what we call reverse cholesterol transport. It drives out to the peripheral tissues, picks up any excess cholesterol lying around, and brings it back to the liver for disposal.
Hence the simple LDL is bad, HDL is good mantra.
It's an oversimplification, but it captures the direction of traffic, yes.
The text goes into a lot of detail on the LDL receptor. It seems like this bad delivery truck has a very sophisticated docking system.
Oh, it's a Nobel Prize-winning mechanism. The cells that need cholesterol express these receptors on their surface. They sit in these little depressions called clathrin-coated pits.
A clathrin-coated pit. It sounds like something from a sci-fi movie.
It's literally an indentation in the cell membrane that's reinforced with a protein scaffold. And sitting in that dock is the LDL receptor, and it's looking for one specific protein on the surface of the LDL particle.
A barcode.
A barcode is a great word for it. It's a protein called ApoB100. When the receptor locks onto ApoB100, it triggers the cell to swallow the entire LDL particle whole.
Endocytosis.
The cell pulls it inside, a lysosome fuses with it, and breaks it down, releasing the cholesterol for the cell to use. And this is a critical part. The receptor itself is then recycled back to the surface to catch another LDL particle.
So the loading dock is reusable.
It's meant to be reusable, unless a protein called PCSK9 gets involved.
Oh, okay. I see ads on TV for PCSK9 inhibitors all the time now. It's clearly the new big thing in cardiology. So what is this protein actually doing?
PCSK9 is basically a receptor assassin. It's a protein made by the liver that gets secreted into the blood. If it binds to an LDL receptor at the same time an LDL particle does, it changes the internal signaling.
How does it change?
When the whole complex is brought inside the cell, instead of the instruction being recycle a receptor, the instruction becomes send the receptor to the lysosome for destruction.
So PCSK9 tells the cell to destroy its own loading docks.
Exactly. And if you have fewer loading docks, your cells can't pull LDL out of the blood efficiently.
So the delivery trucks just keep circling on the highway and the concentration in the blood goes up and up.
That's precisely what happens. And so the inhibitor drugs are antibodies that find and neutralize PCSK9 in the blood. This protects the receptors from destruction, allows more of them to be on the cell surface, and dramatically increases the clearance of LDL from the blood.
It's a brilliant therapeutic strategy. Just protect the loading docks.
It really is.
Okay, so once the cholesterol is inside the cell, it can exist in 2 forms, right? Free and stored. And there are 2 enzymes with very similar names, ACCAT and LCAT. Can you help us keep them straight?
Yes, this is a classic source of confusion. Let's make it simple. ACCAT works inside the cell.
ACCAT, inside. Got it. What does it do?
Free cholesterol is functional, but it's also toxic in high amounts because it makes membranes too rigid. So the cell needs a way to store it safely. ACEAT is the enzyme that attaches a fatty acid to the cholesterol molecule.
It esterifies it.
Yes, turning it into a cholesterol ester. This makes it completely hydrophobic, so it coalesces into a little lipid droplet that can be safely tucked away in the cytoplasm. AT is the warehouse manager, packing away the inventory.
Okay. ACEA is the internal packer. So what is LCAT?
LCAT works outside the cell, in the blood. And it works specifically with HDL, the cleanup crew.
Like a garbage truck.
Right. When an HDL particle goes to a cell and picks up excess cholesterol, which it does via a transporter called ABCA1, the cholesterol is just sitting on the surface of the HDL. It could fall off.
It's not secure.
It's not secure. LCAT is the trash compactor on the truck. It circulates in the blood, finds that cholesterol in the HDL, and esterifies it. This traps the cholesterol in the core of the HDL particle so it can't escape on the trip back to the liver.
I love that. ACEA packs the warehouse, LCAT packs the trash truck.
A perfect way to remember it.
Okay, so the trash truck is full. It's headed back to the liver. This brings us to the final and maybe the most fascinatingly flawed part of this whole system. Section 4, the exit strategy.
Or, more accurately, the profound lack of a robust exit strategy.
What do you mean by that?
This is a really fundamental aspect of our biology. We have this incredibly complex, energy-intensive pathway to build the steroid ring from scratch. We have absolutely no pathway to break it down.
We can't. We can't digest it for energy.
No, we can't catabolize the cholesterol ring structure into carbon dioxide and water like we can with glucose or fatty acids. Once it's made, the core structure is indestructible in our bodies.
So if you can't destroy it, the only option is to excrete it whole.
You have to physically flush it out of the body. And the only way to do that is through the liver, into the bile, and out into the intestine.
And this is where bile acids come in.
This is their starring role. The liver takes cholesterol, and in its peroxisomes, it modifies it into bile acids. The main ones, the primary bile acids, are cholic acid and chenodeoxycholic acid.
Is there a main control point for this process too?
There is. The rate-limiting enzyme is called 7-alpha-hydroxylase, or CYP7A1. It's the first step. And there's a neat little clinical pearl here. The reaction requires several cofactors, including vitamin C.
Wait, so if you're deficient in vitamin C, you could have trouble getting rid of cholesterol.
It's a plausible biochemical link.
Yeah.
If you can't open the exit door efficiently, cholesterol could theoretically back up in the system.
Interesting. So the liver makes these primary bile acids. Then what?
Then it conjugates them. It attaches an amino acid, either glycine or taurine, to the molecule
Why does it do that?
to turn them into better detergents. Pure bile acids aren't soluble enough to work well in the watery environment of the intestine. Adding that amino acid makes them much more effective at emulsifying the fats in your food, breaking big fat globules into tiny little droplets that your enzymes can attack.
So the liver makes these special soaps, dumps them into the gut to help you digest your meal. What happens to them then?
Then, the trillions of tenants living in your gut get involved, the microbiome.
The gut bacteria.
The gut bacteria have their own enzymes. First, they deconjugate the bile acids. They snip off the glycine or taurine we just added. Then they perform another modification. They remove that hydroxyl group at position 7, turning the primary bile acids into secondary bile acids.
So cholic acid becomes deoxycholic acid.
And chenodeoxycholic acid becomes lithocholic acid. The bacteria are chemically altering our waste products.
Okay, so now these secondary bile acids are in the gut. Do we finally just get rid of them?
You would think so. But this is the most counterintuitive and stunning part of the whole story. The body is an extreme miser. It created a system called enterohepatic circulation.
The great recycling loop, the numbers on this in the chapter are just unbelievable.
They're staggering. Your liver secretes somewhere between 15 and 30 grams of these bile salts into your intestine every single day.
30 grams is a lot. That's more than an ounce.
A huge amount. But if you measure how much actually comes out in your feces, it's only about 0.4 grams.
Hold on. 30 grams go in, but less than half a gram comes out. Where did the rest go?
The body reabsorbs it. 98 to 99% of all the bile acids you secrete are actively reabsorbed in the last part of the small intestine, the ileum, and sent straight back to the liver through the portal vein to be used again.
So we use the same biomolecules over and over and over again.
Yes, the liver only has to synthesize that tiny 0.4 grams a day to replace what's lost. It's an incredibly efficient system for conserving a valuable, metabolically expensive product.
But it's also a problem, right? If our goal is to lower body cholesterol, this recycling is our enemy. We want to break the loop.
That's the entire therapeutic principle.
Yeah.
If you can use something to bind up the bile in the intestine so it can't be reabsorbed.
Like a drug or certain types of fiber.
Soluble fiber, exactly. If you force the body to excrete it, the liver panics and says, hey, my bile acid shipment isn't coming back. I have to make more. And what does it use as the raw material to make more?
Cholesterol.
It starts pulling LDL cholesterol out of the bloodstream at a higher rate to feed its bile acid factory. And your blood cholesterol levels go down.
So a high-fiber diet literally forces your body to drain cholesterol from your blood.
That's the mechanism. Now, there's one small exception to the recycling that the text mentions, lithocholic acid.
The secondary bile acid.
Yes. It turns out to be not very water-soluble, so our intestine has a hard time reabsorbing it. A good portion of it actually does get excreted, making it one of the few reliable natural exit routes for cholesterol from the body.
Okay, we've traced the entire life story. We've built it, controlled it, shipped it, recycled it. Now we have to talk about what happens when the system fails. Section 5. Clinical aspects, the big one being atherosclerosis.
The deposition of cholesterol and cholesterol esters from lipoproteins into the walls of arteries, forming plaque.
The source is pretty clear on the correlation. High levels of the delivery trucks, VLDL, LDL is bad. High levels of the cleanup crew, HDL is protective. The ratio is key.
It's all about the balance of traffic. If
Yeah.
you have too many delivery trucks, not enough garbage trucks, trash is going to start piling up on the sides of the road.
Let's get practical and talk about diet. This is where it gets so confusing for people. Saturated fat, unsaturated, trans fats. The chapter actually explains the mechanism of why they matter.
And it's so much more elegant than just fat clogs your pipes. It all comes back to the LDL receptor. It turns out that a diet high in saturated fatty acids causes a downregulation of LDL receptors on the surface of your cells.
So eating a lot of saturated fat literally tells your liver to take down its own loading docks.
Effectively, yes. With fewer receptors, your body can't clear LDL from the blood as quickly, so blood levels rise.
What about unsaturated fats? Olive oil, avocados?
They do the opposite. Polyunsaturated and monounsaturated fatty acids have been shown to induce an upregulation of LDL receptors.
So eating olive oil helps your cells build more loading docks.
That's right. More receptors means faster clearance of LDL from the blood. That's the biochemical reason they are considered heart healthy. They help the logistics system work better.
And then there are trans fats.
The absolute worst of all worlds. They downregulate the LDL receptors, raising your LDL, and they also lower your HDL. So they're blocking the delivery clearance and firing the cleanup crew at the same time. A total disaster for the system.
That's a devastatingly effective way to mess things up. I did see a weird detail in the lifestyle factors section. Drinking hard water versus soft water.
Yeah, it's an old epidemiological observation. Populations that drink hard water, which is rich in minerals like calcium and magnesium, tend to have, on average, slightly lower rates of heart disease than those drinking soft water.
So minerals might be protective somehow?
The mechanism isn't perfectly clear, but it's a consistent finding. It's a minor factor compared to everything else, but it's interesting.
And what about coffee? As a heavy coffee drinker, I'm almost afraid to ask.
The news is nuanced. The source notes an association, but it really depends on how you brew it. The link is strongest with unfiltered coffee methods.
Like a French press or cowboy coffee boiled in a pot.
Exactly. Those methods don't filter out a couple of oily compounds in coffee beans called cafestol and kawel, and those compounds have been shown to raise LDL cholesterol.
But a standard drip coffee machine with a paper filter.
The paper filter traps almost all of it, so your average filtered coffee is probably fine.
Oh, thank goodness. Okay, let's finish up by looking at the genetic lottery. The primary disorders listed in table 26:1.
These are fascinating because they're nature's experiments.
Yeah.
They show us what happens when you break just one specific part of this incredibly complex machine. The classic example is familial hypercholesterolemia.
That's a genetic defect in the LDL receptor, right?
Yes. These individuals are born with faulty or absent loading docks. Their cells are blind to the LDL in their blood. Even on a perfect diet, their LDL levels can be astronomically high, and they can suffer heart attacks in their teens or 20s. It's the ultimate proof of how important that receptor is.
And on the other end of the spectrum.
You have these incredibly lucky people with familial hyperalpha-lipoproteinemia.
That's a mouthful.
It just means genetically high HDL. They're born with an overactive cleanup crew. And as you'd expect, they tend to have very low rates of heart disease and live exceptionally long lives.
Wow. Then you have the really unusual, tragic ones like Tangier disease.
Tangier disease is a deficiency in that ABCA1 transporter.
The little door that lets cholesterol out of the cell and onto the HDL truck.
Right. If that door is broken, the HDL trucks can't pick up any cargo. An empty HDL particle is unstable and gets cleared from the blood very quickly. So these patients have virtually no HDL.
And the cholesterol gets trapped in the tissues?
It accumulates everywhere. Their tonsils famously turn a bright orange-yellow color because they become so engorged with cholesterol esters.
What a bizarre symptom. And fisheye disease.
That's a deficiency in LCAT. The trash compactor on the HDL truck is broken. So the cholesterol it picks up isn't properly stored and it deposits in various tissues, most notably the cornea of the eye, making it cloudy and opaque.
Like a boiled fisheye. The names are very literal.
Sometimes biology is. And the last one to mention is lipoprotein A.
LP(a).
This one is a double threat. It's basically an LDL particle with an extra sticky protein attached to it. So one, it contributes to plaque like any other LDL. But two, that extra protein looks a lot like plasminogen, a key molecule in breaking down blood clots.
So it interferes with clot busting?
Yes, it inhibits fibrinolysis. So people with high LPA not only build more plaque, but if a clot forms on that plaque, their body has a harder time dissolving it. It's a major independent risk factor for heart attacks.
Okay, so we've gone from a single two-carbon molecule all the way to a heart attack. We've seen the factory, the control room, the shipping network, and the clinical consequences. What's the final synthesis? What's the big takeaway from all this?
For me, the overwhelming theme of this entire chapter is that our cholesterol biology is a system designed for an environment of scarcity. Every single mechanism, the expensive synthesis, the 5 layers of regulation, the 99% recycling rate, it all screams, conserve this molecule at all costs.
We are hardwired to hoard it.
Because for almost all of human evolution, getting enough calories and fat was the primary challenge. Cholesterol is precious. You don't throw away something that's so hard to make and so vital for survival.
But now we live in a world of caloric abundance.
We live in a world where that resource is no longer scarce. It's everywhere. Our ancient hoarding biology is now drowning in the very thing it evolved to cherish.
The source has this one line that really stuck with me. It says, humans are one of the few species in which dietary cholesterol induces atherosclerosis.
It's a sobering thought. You can't give a rabbit a heart attack by feeding it a high-fat diet in the same way you can a human. Our machinery is uniquely susceptible.
Which really leaves us with a final kind of provocative thought, doesn't it? Is the #1 killer in the developed world essentially just a massive software-hardware incompatibility? Are we running ancient Stone Age genetic code on modern fast food hardware?
I think that's a brilliant way to frame it. The operations manual written in our DNA hasn't been updated for the current operating environment. It's still optimized for the savannah, not the supermarket.
Well, on that note, I think we'll wrap up this deep dive into Chapter 26. Hopefully you have a newfound respect for the incredibly complex and slightly outdated factory that's running in your liver as we speak.
It's doing its best with the programming it was given.
Thanks for listening, everyone. We'll see you on the next dive.
Take care.